Bismillahirrahmanirrahim. I'm Dr. Zahra Noreen, your course instructor of Plant Physiology 1, BOTN 3119. The topic of today's lecture is about assimilation of sulfur and phosphorus. Objectives of the topic are to enable the students to understand the importance, biogeochemical cycles, and assimilation processes of sulfur and phosphorus. First of all, sulfur. Sulfur is the 10th most abundant element in the universe. It is a brittle, yellow, tasteless, and odorless non-metallic element. The majority of the Earth's sulfur is stored underground in rocks and minerals, including as sulfate salts buried deep within ocean sediments. Sulfur cycle. The process begins with geochemical and meteorological processes, such as weathering of rock. When sulfur is released from rock and comes in contact with air, it is converted into sulfate, which is taken up by plants and microorganisms and converted into organic forms. Animals acquire these organic forms of sulfur from their foods. When organisms die and decompose, some of the sulfur enters the tissue of microorganisms and some is released again as sulfate. There is, however, a continual loss of sulfur from terrestrial ecosystems as some of it drains into lakes and streams and eventually into the ocean as runoff. Additional sulfur enters the ocean through fallout from the atmosphere. Once in the ocean, some of the sulfur cycles through marine communities as it moves through food chains. Some re-enters the atmosphere and some is lost to the ocean depth as it combines with iron to form ferrous sulfide, which is responsible for the black color of the marine sediments. Sulfur re-enters the atmosphere naturally in three major ways. Sea spray releases large amounts of the element from the ocean into the atmosphere. Anaerobic respiration by sulfate-reducing bacteria causes the release of hydrogen sulfide gas, especially from marshes, tidal flats, and similar environments in which anaerobic microorganisms thrive. And the volcanic activity releases additional but much smaller amounts of sulfur gas into the atmosphere. Since the Industrial Revolution, human activities have contributed significantly to the movement of sulfur from the lithosphere to the atmosphere, as the burning of the fossil fuels and the processing of metals have occasioned large emissions of sulfur dioxide. Oxide of sulfur and nitrogen contribute to the acid rain that is the common downwind from these industrial activities. Various pathways of sulfur cycle are shown here. Major sulfur-producing sources include sedimentary rocks, which release hydrogen sulfide gas, and human activities such as smelters and fossil fuel combustion, both of which release sulfur dioxide into the atmosphere. When dissolved in water, sulfur dioxide is hydrolyzed to become sulfuric acid, which is a strong acid and is the major source of acid rain. Sulfur comes back to the soil through dry and wet deposition. Moreover, decomposition of the living organisms also contribute sulfates into the soil. Biological cycle of sulfur. Biological cycle of sulfur means cycling of sulfur through different organisms. Sulfur through biological oxidation by bacteria like lithotrophic and photosynthetic bacteria converted into sulfate. Sulfate in turn is assimilated by plants and bacteria into organic sulfur compounds. In coming slides, we will see in detail how plants assimilate this sulfate obtained from soil into organic sulfur compounds. These organic sulfur compounds can be mineralized by fungi and bacteria into sulfide. This sulfide in turn again converted into sulfate via biological oxidation. The sulfide can also be obtained from dissimilatory reduction of sulfate and sulfur respectively. Importance of sulfur. It is the part of two essential amino acids, cysteine and methionine, structural part of biotin, which is also known as vitamin B7, vitamin H or coenzyme R, for many metabolic processes. It also plays important role in detoxification of heavy metals, as in case of metallothionein and phytogelatin. Phytogelatins are oligomers of glutathione which act as chelators and are important for heavy metal detoxification. As S-adenosylmethionine, which is abbreviated SAM, and S-methylmethionine, SMM, sulfur also play important role as methyl donor in one-carbon metabolism. The catalytic sites of several enzymes and coenzymes such as urease and coenzyme A contain sulfur. Disulfide bridges in proteins play structural and regulatory roles. Sulfur participates in electron transport through iron-sulfur clusters, redox cycles, and oxidative stress, as in case of reduced form of glutathione, GSH. Secondary metabolites products such as glucosinolates, these are the natural chemicals, most likely contribute to the plant defense against pests and disease and impart a characteristic bitter flavor property of cruciferous vegetables. They also have sulfur in their structural formula. The versatility of sulfur derives in part from the property that it shares with nitrogen, that is, the multiple stable oxidation states. As in case of nitrogen oxidation states, ranges from -3 in ammonia to +6 in case of nitrate. As similarly, sulfur also has a wide range of oxidation states ranging from +6 in sulfate to -2 in sulfides. The slide showing the pictorial representation of sulfur importance, showing its involvement in many mechanisms involving the formation of protein, part of vitamin, important part of plant hormones, prosthetic groups, C1 metabolism, and so on. Sulfate is the absorbed form of sulfur in plants. Most of the sulfur in higher plants cells derives from sulfate, which is absorbed by an hydrogen sulfate symporter from the soil solution. Plants can also metabolize sulfur dioxide taken up in the gaseous form through stomata. Nonetheless, prolonged exposure more than 8 hours to high atmospheric concentration, that is greater than 0.3 ppm of sulfur dioxide, causes extensive tissue damage because of the formation of sulfuric acid. Sulfur assimilation. Sulfur in plants are in two forms: organic sulfur, which is obtained in the form of cysteine and methionine, and that is supplied by external sources or internal reserves. Or it can be in the form of inorganic sulfate, which is obtained from the soil and needs sulfate reduction. Firstly, sulfate reduced to H2S and then used to synthesize amino acid cysteine. Sulfate assimilation, that is the reduction of sulfate to cysteine. The first step in the synthesis of sulfur-containing organic compounds is the reduction of sulfate to the amino acid cysteine. Sulfate is a very stable and thus needs to be activated before any subsequent reactions may proceed. Activation begins with the reaction between sulfate and ATP to form 5' adenylyl sulfate, which is sometimes referred to as adenosine 5' phosphosulfate or APS, and pyrophosphate PPI. The reaction is given below. Sulfate plus magnesium ATP give rise to APS and PPI. The enzyme that catalyzes this reaction, that is ATP sulfurylase, has two forms. The major one that is found in the plastids and the minor one which is found in the cytoplasm. The activation reaction is energetically unfavorable. It means to derive this reaction forward, the products APS and PPI must be converted immediately to another compounds. PPI is hydrolyzed to inorganic phosphate PI by inorganic pyrophosphatase according to the following reaction, in which PPI plus water give rise to 2 molecules of PI, inorganic phosphate. The other product APS is rapidly reduced or sulfated. Reduction is the dominant pathway. Reduction of APS. The reduction of APS is a multi-step process that occurs exclusively in the plastids. First, APS reductase transfers 2 electrons apparently from reduced glutathione GSH to produce sulfite SO3-2. APS plus 2 molecules of glutathione in reduced form give rise to 1 molecule of sulfite plus 2 hydrogenine plus 1 molecule of glutathione in oxidized form plus 1 molecule of AMP, where GSSG stands for oxidized glutathione and GSH represents reduced glutathione. The SH in GSH and the SS in GSSG stands for sulfur hydrogen and sulfur-sulfur bonds respectively. Second, the sulfite reductase transfers 6 electrons from ferredoxin to produce sulfide. Sulfite plus 6 molecule of ferredoxin reduced form give rise to 1 molecule of sulfide plus 6 molecule of ferredoxin oxidized form. The resultant sulfide then react with O-acetylserine to form cysteine and acetate. The O-acetylserine that reacts with sulfide is formed in a reaction catalyzed by serine acetyltransferase. Serine plus acetyl-CoA give rise to O-acetylserine plus coenzyme A. The reaction that produces cysteine and acetate is catalyzed by O-acetylserine thiolase. Now, sulfation of APS. Sulfation of APS localized in the cytosol, and it is an alternative pathway to reduction of APS. First, APS kinase catalyzes a reaction of APS with ATP to form 3' phosphoadenosine 5' phosphosulfate, that is PAPS. APS plus ATP give rise to 1 molecule of PAPS plus 1 molecule of adenine diphosphate ADP. Sulfotransferases then may transfer the sulfate group from PAPS to various compounds, including choline, brassinosteroids, flavanol, gallic acid glucoside, glucosinolates, peptides, and polysaccharides. The slide is presenting structure and pathways of compounds involved in the sulfur assimilation. First of all, the sulfate is assimilated into sulfide through a series of reactions, which is then utilized for the formation of cysteine, which in turn utilized for the formation of sulfur-containing compounds. Sulfate, first of all, with the help of ATP sulfurylase enzyme, which cleaves the PPI pyrophosphate from ATP and replace it with sulfur, make APS, adenosine 5' phosphosulfate. After the formation of APS, the APS has two choices. One is its reduction, which will form sulfide from it, and the other one is the sulfation, which takes place in the cytosol, as I told you in the previous slides. Sulfation will result in the formation of PAPS with the help of enzyme APS kinase. PAPS then get transferred into O-sulfated metabolites, which is utilized for the formation of choline, brassinosteroids, flavanol, gallic acid glucosides, glucosinolates, peptides, and polysaccharides. In the case of reduction which takes place in plastids, APS gets transferred into sulfide through a series of reactions in which the reduction takes place with the help of glutathione and ferredoxin. Sulfide is then react with O-acetylserine to form cysteine. Cysteine and then utilized for the formation of other organic compounds which contain sulfur. The current slide is presenting the structure formula of oxidized and reduced glutathione. Glutathione is a high-energy cofactor of glutathione transferase enzyme, and it has two forms. One is reduced glutathione, and the other one is oxidized form of glutathione. Reduced glutathione is abbreviated as GSH, while the oxidized glutathione is represented as GSSG. As you can see, glutathione is made up of three amino acids: glycine, cysteine, and glutamate. So actually, the cysteine is the amino acid which is responsible for its two forms, the reduced form and the oxidized form. In case of reduced form, sulfur hydrogen bond is present, and in case of oxidized form, sulfur-sulfur bridge is present between the two parts of cysteine of glutathione. Site of sulfur assimilation in plants. Sulfate assimilation occurs mostly in leaves. To a lesser degree, volatile sulfur compounds such as carbon disulfide, carbonyl sulfide, hydrogen sulfide, methyl mercaptan, and sulfur dioxide can be taken up by leaves. The magnitude of the flux of gaseous sulfur compounds into the plant leaves has been ranked in the following order: sulfur dioxide greater than hydrogen sulfide is greater than carbonyl sulfide, greater than methyl mercaptan, and greater than carbon disulfide. In order to get assimilated, the reduction of sulfate to cysteine changes the oxidation number of sulfur from +6 to -4, thus entailing the transfer of 10 electrons. Glutathione, ferredoxin, and NADPH or O-acetylserine may serve as electron donors at various steps of this pathway. Leaves are generally much more active than roots in sulfur assimilation, presumably because photosynthesis provides reduced ferredoxin and photorespiration generates serine that may stimulate the production of O-acetylserine. Sulfur assimilated in leaves is exported via the phloem to the sites of protein synthesis that may be shoot, root apices, and fruits, mainly as glutathione. Methionine is synthesized from cysteine. Methionine, which is the other sulfur-containing amino acid found in proteins, is synthesized in plastid from cysteine. After cysteine and methionine are synthesized, sulfur can be incorporated into proteins and a variety of other compounds such as acetyl-CoA and S-adenosylmethionine, that is SAM. SAM is an important in the synthesis of ethylene, which is an important plant growth regulator, and in reactions involving the transfer of methyl groups as in lignin synthesis. Here is the pathway of modification of cysteine to methionine. Cysteine plus O-phosphohomoserine with the help of enzyme cystathionine gamma synthase will be converted into cystathionine. Cystathionine will cleave out a pyruvate plus NH3, and in the presence of enzyme cystathionine beta lyase will be converted into homocysteine. Homocysteine with the addition of one methyl group in the presence of enzyme methionine synthase will be converted into methionine. Sulfur uptake and metabolism. Here is summary of sulfur uptake and metabolism in plants. Sulfur is actively taken up by root cells in the form of sulfate. Sulfate is passed through the root epidermis and cortex up to the xylem and translocated into the shoot and the leaves, as shown in figure of coming slide. In the leaves, sulfate is activated in the cell. The activated sulfate is reduced to the level of sulfide. Reduced sulfate is necessary for the synthesis of the sulfur-containing amino acids like cysteine and methionine, the important biological tripeptide glutathione, and finally of proteins. It is also necessary for the biosynthesis of sulfolipids, which are important components of biomembranes. As soon as there is a surplus of activity, here is the pictorial representation of this summary in which you can see the sulfur can be assimilated, can be incorporated via leaves, and it can be taken up via roots. In case of roots, it will be transported to the leaves through xylem, and then after the transportation, it gets assimilated via a series of reaction into the sulfur containing amino acid cysteine. Another way out is the accumulation of the sulfate or the sulfate containing compounds into the vacuole. After the formation of cysteine, it can be converted into methionine, or it can be utilized for the formation of glucosinolates, secondary metabolites, or glutathione, which is the transported form of sulfur in plants. Glutathione can be utilized for the formation of phytochelatins, and it can be retranslocated via phloem into the root. In the roots, it again can be converted into an amino acid cysteine, which is utilized for the formation of proteins, or it can be utilized for the formation of phytogelatins. Here is the putative connection of nitrate and sulfur assimilation via O-acetylserine. On the left side of the pathway is nitrate assimilation, and on the right side of the pathway is sulfate assimilation. Sulfate assimilation gives rise to the formation of cysteine, and the nitrate assimilation gives rise to amino acid like serine, which after being converted into O-acetylserine can be utilized for the formation of amino acid cysteine, which is sulfur containing amino acid, which is then utilized for the formation of protein and a number of secondary metabolites containing sulfur. The rest of the story is discussed in the previous slides. Now the importance biogeochemical cycle and assimilation of phosphorus. Most of the phosphorus occurs as phosphate. Phosphate in the form of HPO4 in the soil solution is readily absorbed by plant roots via a hydrogen phosphate symporter and incorporated into a variety of organic compounds including sugar phosphates, phospholipids, and nucleotides. It is often limiting in the aquatic environments because in aerobic water it gets precipitates and is lost as sediments. Phosphorous cycle. The phosphorus cycle differs from the other major biogeochemical cycles in that it does not include a gas phase, although small amounts of phosphoric acid may make their way into the atmosphere contributing, in some cases, to acid rain. The water, carbon, nitrogen and sulfur cycles all include at least one phase in which the element is in its gaseous state. Very little phosphorus, however, circulates in the atmosphere, Because at Earth's normal temperatures and pressures, phosphorus and its various compounds are not gases. The largest reservoir of phosphorus is in sedimentary rock. Overall, you can divide phosphorus cycle into three parts. The phosphorus cycle is representing here. Actually, it involves two mini cycles. One is operating in the soil and the other one is operating in the aquatic environment. The three parts, as I told you in the previous slide, can be seen here. Reservoir, take up by the autotrophs and then return to the reservoir. The same is the case of the aquatic environment, in which the main reservoir is the dissolved phosphorus in ocean water, which is taken up by the autotrophs of the aquatic environment, and then through the marine food webs, it again enters into the water. Other pathways which add the phosphorus to the reservoirs include the excretion in the form of guano. The withering of the guano adds more phosphorus into the aquatic environment. Furthermore, the use of fertilizers due to the human activity also add more phosphorus to the land. A link of phosphorus cycle is given. You can visit this link for the further clarification of phosphorus cycle.
The last of the biogeochemical cycles we'll be talking about this chapter is the phosphorus cycle. I like to do this one last because in a lot of ways this is the simplest cycle that we've talked about so far. The reason this one's easy is if you look up here at the atmosphere part of the cycle, there are no arrows coming towards or leaving from the atmosphere, which means there is no atmospheric stage to the phosphorus cycle. Everything is taking place either in the soil or in the water, somewhere on the ground that keeps it out of the atmosphere. And I think the complicated parts of the nitrogen cycle and the carbon cycle are what moves those two particular elements to or from the atmosphere. So the key to this one is this stuff. It's all in, as it calls in this diagram, the mineable rocks. It's all in the minerals. There's two ways that phosphorus gets out of the minerals. One of the ways is weathering, the other one is mining. The weathering idea could be erosion, anything that's breaking down those rocks and making the phosphorus available. The material that's weathered out of the rocks is either going to go into rivers or directly into the soil. When it's in the soil, it gets uptake into land plants, and then those plants are eaten by consumers. So that's ultimately the way that we get all of the phosphorus that we need. It comes from the vegetables and things that you are consuming. Those consumers then can eventually decompose and go back to the soil, and that kind of creates its own little mini cycle over on the side. It's the same general approach in the ocean. The producers are getting it from the water itself, and then they're eaten by consumers, which eventually die, go down to decomposers, and that keeps on going in its own little cycle as well. So the phosphorus cycle is ultimately pretty simple compared to the other two. Just like in the previous cycles, we always have to talk about human impact as the last stage. You can see here fertilizers and detergents is the main way that people impact this cycle. If you use dish detergent, laundry detergent, things like that at home, that contains a lot of phosphorus in it. And that eventually has to go through a wastewater treatment plant in order to get removed from the water before that water can be released into the environment. Like anything else, too much of a good thing becomes a bad thing. Phosphorus is required for plants to be healthy, but a lot of it really allows algae to take off and do very, very, very well. So well, actually, that it can deplete a lot of the oxygen from an environment because it's thriving so much. So phosphorus is one of those things that people are producing in high amounts, but we do have to be careful about how much of it we're releasing into the environment, because it is something that should be released slowly. If you think about how it's originally getting there naturally, it's coming out of these mineable rocks, it's coming out of minerals, basically, that are broken down through weathering. So as people go through and release things faster, we kind of unbalance this cycle a little bit. Overall, I think this is probably the simplest one, just because, again, there is none of that atmospheric stage in the phosphorus cycle. So this concludes our study of the biogeochemical cycles. Remember, we have water, carbon, nitrogen, and phosphorus. As always, thank you for watching.
Importance of phosphorus. Phosphorus is an important element for all forms of life. As phosphate, it makes up an important part of the structural framework that holds DNA and RNA together. Phosphate are also a critical component of ATP, the cellular energy carrier for organisms to use in building proteins or contracting muscles. Phosphorus is an integral component of important compounds of plant cells, the sugar phosphate intermediates of respiration and photosynthesis. It is also the part of phospholipids that make up plant membranes. Like calcium, phosphorus is important to vertebrates as well. In the human body, 80% of phosphorus is found in teeth and bones. Phosphate assimilation. The main entry point of phosphate into assimilatory pathways occurs during the formation of ATP, the energy currency of the cell. In the overall reaction for this process, inorganic phosphate is added to the second phosphate group in adenosine diphosphate to form a phosphate ester bond. Types of phosphorylation. There are three types of phosphorylation. 1. Oxidative phosphorylation. 2. Photophosphorylation. And number 3. The substrate level phosphorylation. Oxidative phosphorylation. In mitochondria, the energy for ATP synthesis derives from the oxidation of NADH and FADH by oxidative phosphorylation. It is a typical example of ETC electron transport chain. Photophosphorylation occurs in case of chloroplast. Here, ATP synthesis is also driven by light-dependent photophosphorylation in the chloroplast. Substrate level phosphorylation. In addition to these reactions in mitochondria and chloroplasts, reactions in the cytosol also assimilate phosphate. Glycolysis incorporates inorganic phosphate into 1,3-bisphosphoglyceric acid, forming a high-energy acyl phosphate group. This phosphate can be donated to ADP to form ATP in a substrate-level phosphorylation reaction. Once incorporated into ATP, the phosphate group may be transferred via many different reactions to form the various phosphorylated compounds found in higher plant cells. Here are some suggested books and research articles to further enhance your knowledge about the topic. This is all about today's lecture. Thank you very much. If you have any questions,